it's a pleasure to be here. So yeah, hi, I'm Ed Steele. I'm one of the co-founders of Hoxton Farms. So the world is hungry. The meat industry is fundamentally broken. It's a huge industry, but everyone here in particular knows how it's killing us and it's killing the planet. At the same time, demand for plant-based meat is soaring. But plant-based meat still has a long way to go if it's going to replicate and outperform meat in every dish from every recipe all over the world. Plant-based meat still has a big fat problem. At Hoxton Farms, we grow real cell-based animal fat without the animals. We'll be selling our cultivated fat as a B2B ingredient to the meat alternatives industry, starting with plant-based meat, so that we can finally make meat alternatives that look and cook and taste just as good as the real thing. But why fat? At Hoxton Farms, we're obsessed with fat, and with good reason. It's the single most important sensory component of the meat that we eat. Whether it's prized cuts like ham on Iberico or Kobe Wagyu beef, or the more prosaic like a hamburger or chicken breast, it's the fat that makes meat delicious. We've shown that adding just a small amount of real animal fat to plant-based protein totally transforms the product. And our early customers, the plant-based meat companies, tell us just how desperately they need a replacement for plant oils. And that's where we come in. To do it, we take a harmless handful of cells from an animal and grow them in a bioreactor. We differentiate the cells into fat tissue and we harvest it at the end. We've been hearing a lot about hybrid products today. Our fat can be used alongside plant proteins or cultivated muscle. So who are we? We're based in Old Street in London and have been going for just over a year. We're building an amazing interdisciplinary team of stem cell biologists, engineers, food scientists. I'm a mathematician by background, and my co-founder Max is a synthetic biologist. What we do combines my world of machine learning and optimization with his world of synthetic and molecular biology to use computational techniques that redefine the way that we grow stem cells for cell ag. I actually really don't like this slide because it's our awesome team who do all of the work, and I get to come up here and talk about it. Everything here is the product of hard work from a really talented group, and it's a privilege to work with them every day. There are so many technical challenges associated with growing cultivated fat at scale, which we can broadly fit into 4 categories: cell lines, culture media, bioprocess design, and food science. We use mathematical modeling to help us solve challenges in all of these categories. As a mathematician, I see all of these challenges as optimization problems. We have lots of levers we can pull to affect how the cells grow, the yield we get from our bioprocess, and the type of fat that we make. We want to find the best possible combination of levers in order to make a product that can truly scale. When we break it down like this, mathematically, it sounds simple. The challenge is that there are so many levers. There's cell lines, media components, which can change over time, bioreactor designs, etc., that even if we consider each of these independently in turn, we have a huge space of possible combinations to try. Actually, the biggest problems come from the ways in which each of these areas interact with one another. They aren't independent. The choice of the starting cells affects what media we should use, how we proliferate our cells affects how they differentiate, and the choice of scaffold needs to be jointly optimized with the bioreactor design. Our model has to take into account all of these together to get a solution that's globally optimal, which is really tough. And with that caveat, I'm going to dive into one particular way that we can use data and machine learning to optimize one part of the process, culture media for proliferation. As we all know, culture media is one of the most expensive parts of the process, and the industry needs cheap food grade alternatives to FBS. Some great companies are working on reducing the cost of producing growth factors. I'm going to focus on how we can build a feedback loop for culture media optimization to help us determine which components to use and how much of those components. I'll start off with a toy example, really. Suppose our culture media is made up of two things, water and one growth factor. And we want to know what concentration of the growth factor is best. First off, we need to define what we mean by best here. In our toy example, let's say that there are two things really that are important. The growth of our cells after 3 days, say, and the cost of the mixture. There are loads of possibilities for the function that we're trying to optimize could be. But let's suppose that we want to maximize the growth per dollar of media. With only one component, that means just the growth rate divided by the cost of the growth factor. We could just try a bunch of different concentrations and pick whichever performs the best out of that group. But the problem is that won't scale as we introduce new components. The space of possible combinations will explode combinatorially. To explain what I mean by that, suppose that for each ingredient, there are 20 possible concentrations that we could use. For one component, we have to do 20 experiments to try all of them. But every time we add a new ingredient, we multiply this number by 20. So with 5 possible ingredients, we have 3 million experiments. 3 times 10 to the 6 is 3 with 6 zeros after it. With 20 possible ingredients, we have 1 times 10 to the 26, so 1 with 26 zeros after it. That's how many experiments we have to do. To give you an idea of how big a number that is, it's 5 orders of magnitude larger than the number we would get if we counted all of the cells in all of the cows on Earth. So we need a much cleverer way of searching through this space of possible combinations. Instead, we use an algorithm to do so. We build a feedback loop where we generate data from experiments in the wet lab. We feed those into a model. The model then suggests more experiments to do in the wet lab, and that process continues and continues. That means that we continually improve over time the media recipe that we develop in a really systematic way. I'll demonstrate this for our toy example with only one growth factor, so it's clear, but we can extend the same technique for any number of ingredients. To start off with, we don't have any data on what concentration of the growth factor we should use, so the model doesn't have anything to go on. So we seed the model by trying a few random concentrations. These grey dots are what we observe experimentally. The x-axis shows the different concentrations we're testing in the wet lab, and the y-axis is the growth per dollar of media. So we now want to ask the model 2 questions. Firstly, what do you predict the best concentration is for this growth factor? And secondly, what concentration should we try next experimentally? To answer the first question, we build a machine learning regression model which predicts the growth per dollar of media at every possible concentration based on the data that we have so far. There are pretty standard techniques for doing this, but part of the complexity of the approach involves choosing the right way to build this underlying prediction. What's really important is that the model is Bayesian. It has a concept of uncertainty in the prediction that it makes. So in other words, for every concentration, the model outputs not just a value for the growth per dollar of media, but also another value for how certain the model is in the prediction that it's making. So this green line is the model's prediction for every concentration. As I said, the model also outputs its confidence in the prediction for each concentration, but I haven't drawn this here. We can see that the model predicts that the concentration, denoted by this yellow line, will result in the highest growth per dollar of media. But in this area, the model has the least confidence about its predictions. That's because the nearest points that we've tried so far are quite far away from this area. The model combines both of these to decide which experiment we should try next. On the one hand, it wants to exploit the area that it thinks will have the highest growth per dollar of media. But on the other hand, it wants to explore areas where it has little confidence in the predictions that it's making. This is a trade-off of the model needs to make. And we aim to ensure that we don't get stuck only looking at areas that might not be optimal. We want to explore as much of the space as possible, but focusing and exploiting the areas where we think it's likely that there'll be the highest growth per dollar of media. In this case, our model moves slightly right of the predicted maximum and suggests we try a higher concentration at this new yellow line. So the model has done the trade-off between the exploration and exploitation, and this is the concentration that it suggests we try next experimentally. So we go to the wet lab and we try that new point, and it actually gives us a growth per dollar of media lower than our highest value so far. So we can see the new grey dot here. But we're now back in a position of having a set of data points. So we train the model on the data that we had before and the result of this new experiment. The model makes a new set of predictions for every concentration, which is the new green line. Again, the model has a trade-off between exploitation and exploration, and it suggests a new experiment denoted by this yellow line. So again, we perform this experiment in the wet lab and feed the result back into the model. The model makes a new prediction and the process continues over and over again. And with this feedback loop, we keep going around this flywheel and getting better and better until we find a point where we have the concentration of the growth factor that maximizes the growth per dollar of media. And that's the point that you can see here. With one media component, this example is a little bit contrived. But as we've said, with more ingredients, it becomes impossible to use combinatorial search to find the optimal media recipe. It sounds great, but why is this hard? The more media components we have, the larger our space gets, and the harder the model finds it to suggest the best candidates. This is a great framework, but we have to be clever about how we build the underlying predictions, what function we want to optimize, and how we incorporate other variables into this as well. This is just a small example of a fraction of the modeling work that we do at Hoxton Farms, but hopefully it gives you a bit of a flavor of how machine learning works and how it can be used in cell ag to improve the bioprocess. I've brought this slide up again to remind you that there are multiple places where we use mathematical modeling. And one of the key challenges is how we bring all of these together into one overall framework. If you want to learn more about our modeling work, come and work for us. We're hiring computational biologists, software engineers, machine learning researchers, and of course, wet lab biologists and bioprocess engineers who work really, really closely with the modeling team. You can visit our website here or feel free to get in touch. Thanks for listening. Happy to answer any questions.